The video highlights the essential roles of pharmacology and toxicology studies, illustrating how these animal studies build a crucial foundation for understanding medical products prior to entering clinical trials. In this context, we recognize the invaluable insights gained from safety assessments. Continuing this discussion, we delve into the overview of chemistry and manufacturing controls, or CMC, detailing their regulatory importance and the dynamic evolution they undergo throughout the clinical development phases. This ensures that investigational products maintain consistent quality. To further our understanding, we explore the regulatory definitions surrounding drug substances and products, emphasizing the critical nature of stability and quality control measures in the context of IND submissions. Such diligence safeguards the integrity of clinical studies. Moreover, regulatory submissions must encompass comprehensive information on impurities and the distinctions between clinical and toxicology batches, allowing for a thorough assessment of safety during drug development. This commitment to quality ensures effective evaluation of drug properties. The review process necessitates collaboration across various departments to evaluate molecular variability and the manufacturing methods of drug substances. This multidisciplinary approach enhances regulatory oversight and promotes rigorous safety standards. Characterization of drug substances is pivotal, encompassing both structural and physical chemical data critical for confirming the identity and understanding the behavior of drugs in formulations. Comprehensive data submissions facilitate effective and streamlined reviews. Understanding potential sources of impurities in drug manufacturing processes is essential for maintaining the quality of the final product. Early identification of these impurities can significantly mitigate safety risks for patients. The specification table serves as a vital guide for testing and releasing clinical batches, ensuring adherence to regulatory standards. This table delineates test methods and acceptance criteria, reinforcing our commitment to quality assurance. Stability studies emerge as critical assessments, determining how drug quality evolves over time and impacting both shelf life and retest periods. Through these studies, we evaluate the influence of environmental factors on drug substances. In addition, drug product specifications must encompass identity, strength, quality, purity, potency, and bioavailability, along with clearly defined analytical procedures and acceptance criteria. This thorough approach ensures that products meet stringent safety standards. Equally important is the stability data, which is fundamental in ascertaining shelf life and expiration dates, necessitating rigorous testing under diverse storage conditions. Indeed, preliminary stability information plays a crucial role in initial IND submissions. Clear and effective communication with the FDA through pre-IND meetings proves invaluable in addressing potential CMC issues and regulatory concerns before submission. This proactive stance is vital for paving a successful path in drug development. Pharmacology studies critically evaluate the drug's mechanism of action and its activity in both in vitro and in vivo environments, providing essential data that underscore drug efficacy. This aspect is particularly crucial when evaluating oncology products. Safety pharmacology studies delve into evaluating potential adverse effects on vital organ systems, thereby aiding in identifying risks associated with the drug. These assessments encompass vital areas, including cardiovascular and central nervous system functions. Furthermore, pharmacokinetic studies elucidate how a drug is absorbed, distributed, metabolized, and excreted within the body. Understanding these pharmacokinetic parameters is essential for determining appropriate dosing regimens and establishing safety profiles. It is also crucial to recognize that differences in drug exposure between male and female animals can significantly influence the determination of a safe clinical starting dose. This understanding directly impacts the overall efficacy of the drug. Toxicology studies play a pivotal role in identifying potential dose-limiting toxicities, which are crucial for clinical safety monitoring and may expose toxicities that remain undetected during clinical trials. To ensure comprehensive safety data is available for informed decision-making, the duration of toxicology studies must align with the length of clinical trials. This synchronization is critical for patient safety. In the context of drug development, kidney toxicity became a significant concern during the progression of an antibody-drug conjugate. This led to pivotal modifications in dosing and monitoring protocols, emphasizing the necessity of patient safety. Moreover, genetic toxicology assessments are indispensable in evaluating the potential carcinogenic effects of drugs. These evaluations inform clinical trial designs and patient safety measures throughout the entire development process. In fact, reproductive toxicology studies have now become standard practice in drug development, helping to avert risks akin to those observed with thalidomide, which infamously resulted in severe birth defects. Safety pharmacology encompasses core system assessments, including cardiovascular and respiratory functions, ensuring that drugs do not elicit harmful effects. Grasping these systems is paramount for ensuring patient safety. Genotoxicity studies must be thoroughly completed prior to clinical trials to evaluate potential genetic risks associated with drugs. This vital step safeguards the safety of drugs intended for human application. Clinical pharmacology assumes a critical function in establishing the dosing regimen of a drug, ensuring that the right medication is administered at the correct dosage and timing for patients. Moreover, measuring unbound drug levels is essential for accurately interpreting pharmacological activity, as only free drugs exert therapeutic effects. This crucial measurement serves as a guide for effective clinical decisions. Drug elimination encompasses both metabolism and excretion, which are vital to assessing how the body processes a drug. Key factors, such as hepatic metabolism and renal excretion, play significant roles in this process. Beginning clinical pharmacology studies as early as possible in drug development is instrumental, as they provide critical safety and tolerability data that can influence subsequent phases. Early insights dramatically enhance the likelihood of success. Food effects studies play an integral role in determining the optimal administration of drugs and guiding labeling instructions related to food intake. These studies are essential for maximizing drug efficacy during clinical trials. Furthermore, hepatic and renal impairment studies are critical in assessing drug metabolism and elimination. The FDA provides comprehensive guidelines regarding the timing of these studies to ensure patient safety is upheld. Drug interaction assessments necessitate thorough in vitro and in vivo testing of investigational drugs. These studies not only inform drug labeling but also identify potential risks associated with combination therapies. The significance of clinical trial simulation cannot be overstated, as it aids in estimating dose selection and predicting trial outcomes with greater accuracy. This strategic approach significantly enhances the efficiency of trial processes. Moreover, predictive safety evaluations utilizing systems pharmacology can identify critical safety biomarkers, thus minimizing risks associated with new drug formulations and fostering a safer therapeutic landscape. When introducing new formulations during clinical trials, the careful timing of protocol submissions and CMC information is paramount. Effective coordination between these submissions is essential for ensuring regulatory compliance. Conducting food effects studies becomes even more crucial when a novel drug formulation is developed, as the composition of food can alter the pharmacokinetics of the drug. This meticulous attention to detail ensures accurate dosage and enhanced effectiveness. In addition, regulatory bodies require comprehensive batch data to thoroughly assess the safety and efficacy of drug products, with a focused evaluation on differences in impurity profiles between batches. This diligence is fundamental for informed approval decisions. The determination of starting doses for immunotherapies hinges on an understanding of minimal anticipated biological effect levels, integrating data from pharmacology and toxicology studies. This foundation plays a crucial role in ensuring patient safety throughout clinical trials. Moreover, in vitro screening is integral to identify any interactions a drug may have with drug metabolizing enzymes. Positive results from these screenings pave the way for further clinical trials that delve deeper into evaluation. Common CMC issues that arise during FDA reviews often encompass safety concerns related to drug identity, purity, strength, and overall quality. Addressing these factors comprehensively is crucial to avert clinical holds and ensure successful trial progression. Food effects studies are recommended early on in drug development to assess the interplay between food and drug concentration. This essential knowledge is critical for providing patient instructions and for planning future trials effectively. It is noteworthy that higher-order mammals, such as dogs and monkeys, may exhibit varying sensitivities to drugs, which can influence the determination of clinical starting doses. This observation underscores the necessity of incorporating diverse species within toxicology studies. Lastly, biological pharmaceuticals frequently necessitate the inclusion of non-human primates as pharmacologically relevant species for toxicology studies, reinforcing the importance of ensuring both safety and efficacy in new drug developments. Finally, adherence to good laboratory practice, GLP principles in toxicology studies is imperative, ensuring reliability and safety throughout the drug development process. This compliance stands as a cornerstone for conducting pivotal toxicology studies.